In this video today, we are going to talk about the Lineweaver-Burk plot and what it's all about. Before we learn about the Lineweaver-Burk plot, we'll need to have an understanding of the Michaelis-Menten plot. So let's take a look. The Michaelis-Menten plot is a graph that represents velocity, which you can say is how fast the reaction goes in relation to the substrate concentration. It's used to plot three different enzyme catalyzed reactions, the first being the control. The second is competitive inhibition. This is where there's an inhibitor that blocks the substrate from binding to the enzyme. However, some of the substrate does bind but requires a much higher concentration of the substrate in order to proceed. The third is non-competitive inhibition. This is where an enzyme contains an allosteric site where an inhibitor can bind. This results in a conformation change of the enzyme affecting the substrate's ability to properly bind to the enzyme. Each one of these reactions aims to reach Vmax. The control and competitive inhibition are the only ones able to do this effectively. The difference between the two, however, is that competitive inhibition requires a much higher substrate concentration and therefore alters the Km value, which is found by finding what the half Vmax is and translating that to an X value. This plot can be represented by this equation. Even though this plot is a very efficient plot, it has its limitations. The first being that the curve never actually reaches Vmax. With this, if the Vmax is uncertain, the Km is also uncertain, as well as an approximate value. Now that we know a little bit about the Michaelis-Menten plot, we can finally get into the fun stuff. So here we go. The Lineweaver-Burk plot takes each enzyme catalyzed reaction from the Michaelis-Menten plot and turns those curved lines into straight lines. This is done by extrapolating the curved lines and plotting these points as a straight line. This gives a better visual of the Km values. This plot can also be represented by this equation. Now, if we recall the equation in the Michaelis-Menten plot and take the reciprocal of both sides, also known as a double reciprocal, we get the equation of the Lineweaver-Burk plot. This equation represents the equation of a line, y equals mx plus b, where y equals 1 over v, m, which is the slope, equals km over vmax, x equals 1 over the substrate concentration, b, which is also the y-intercept, equals 1 over vmax, and the x-intercept equals negative 1 over km. Now let's have a look at some of the advantages to this plot. This Lineweaver-Burk plot connects the curved lines in the Michaelis-Menten plot and forms a straight line. This makes it easier to visualize. This plot also gives the direct determination of Vmax and Km from the x and y intercepts of the graph. It's also a good representation when comparing different types of enzyme inhibition, like competitive and non-competitive inhibition. Now, no plot is perfect, so we do have to look at some of the disadvantages of this plot. The Lineweaver-Burk plot presents an unequal weighting of data because low substrate concentration, which is also large reciprocal substrate concentration, dominates the majority of the plot and gives more notice to experimental error. This plot also requires extrapolation, which can cause the Vmax and Km to be outside of the values measured. The Lineweaver-Burk plot is also often less accurate than other nonlinear and linear representations. Because of all these, this plot is rarely used in enzyme kinetics and is not the most accurate representation of what is going on. Well, there we have it. Now you know a little bit about enzyme inhibition and the Michaelis-Menten plot. Even better, you now know what the Lineweaver-Burk plot is and how it's used in enzyme kinetics.